Welcome to this introductory video on our course I1 on bioimage analysis. So for this course, we're having two labs on the 5th and the 12th, with some time in between before and after to kind of engage with the different material that's on Absorb and also the end drive. There's also a range of resources that you use on the different packages that we're using, such as Collocar, Bioimage. There's videos on these and the general principles behind bioimage analysis, including other packages such as Magic, which we're not really covering in this course. And there's also resources here on R on Bioconductor about Imager, which we're using as well as Bioimage. There's also example papers here on African swine fever virus, and then also viruses, on how imaging data can be used. And then also another example paper on using the Amaris software that we're actually not directly using in this course because we're using RStudio to achieve this. However, it's the same sort of principle to achieve that using that software tool. So what we want to do is learn key concepts in image analysis and then also use R as a key tool to perform standard image analysis, particularly those relating to confocal microscopy. So what we're going to consider is taking files in and out, image files in and out, using different manipulations to, say, perform thresholding or segmentation, and then try to identify features that are of interest with R. So what we want to be able to do by the end of this course is be able to load, filter, transform images with R, and particularly in relation to confocal microscopy, to be able to understand particular images in that manner. So what we're kind of thinking more broadly on this course is how we interpret images. And images are kind of fundamental to our daily lives, as well as our scientific ones. So even if you don't handle bioimage data directly, you'll have many experiences interpreting images in different ways, and how objective measurements using different software tools or coding environments can help us achieve that. Images are particularly important, and they can have many different applications, such as epidemiological maps for combating disease to identify the sources of different diseases. And there's also more broad considerations of, say, taking satellite data of Earth to look at climate change related phenomena. So we can think about our microscopy approaches on many different levels. We can also think about how the images are visually comprehensible. So people might have red, green color blindness, and then also how different images can have identify different features in different ways, and tell us more about what's going on. So you can see that the images being shown here are all quite different, have different meanings, and those meanings are interpreted by the person viewing them, and that's kind of similar to what we are doing in a sense. We're trying to make interpretable images that may have meaning for the viewer. There's huge interest in images. So this is the top 10 most liked images from Instagram overlaying on top of each other. So you can see when you do that sort of thing, you see quite a complex landscape where there's different features. If you look at it in different ways, you can see different things. And that's like a lot of microscopy images. We know that human input is essential, because if you think about asking a computer to do certain things like distinguish between a dog and a muffin, they can struggle with this task. For humans, it's pretty easy because we have a whole series of features that we use to distinguish between A and B, whereas automating this is more challenging. And you can think of simple examples where it becomes actually quite difficult for computers to achieve what humans find very, very easy. And there's copious examples of that, where you have different dog breeds and chicken nuggets here, or fried chicken, should I say, that it's easy for us, whereas trying to do that with tools requires optimization, consideration, and particular parameters that are specific to the experiment at hand. And if you do the same experiment somewhere else, you'll get different results, and therefore it's easy for us to identify maybe what it is, but it isn't always obvious for someone without prior experience. So creating good images takes time and effort and is not difficult to do, but can be really informative when we achieve that goal. So the example images are listed here. So this is some bronchitis viruses in chicken trachea here in this first image. So you can see kind of the cell membranes, the villi and everything along here. Good example of African swine fever viruses shown by the kind of darker areas there. And you think about how you identify how many viruses are present, how to divide that between the cells. And then also we have example of a negative stain to show blue tongue viruses here, where you can see actually really high resolution to different virus particles. There's other examples here of chicken gut epithelium. We also have an African swine fever infected cell. And then on our right, we have foot and mouth disease virus and infected tongue epithelium. There's more of the same tongue epithelium here as well. And we also see over here smooth endoplasmic reticulum in this very, very high resolution microscopy image here in the middle. And then we also have planet cells over here on the right too. More examples include uninfected cells on the right that we can compare to ones that are infected. We have a Vimentin labeled cell here, and then cells expressing African swine fever protein as well on our right. So you can think about, in terms of bioimage data, what are your favorite figures? Why are they useful? And what do you think is best if you're trying to represent image data for others? What do you think works well? So all those are examples of things that we'll explore subsequently in this course.